In this lecture, our last lecture on blood, we will discuss blood groups and transfusions. Now, blood is vital for transporting substances through the body. When blood is lost, the blood vessels constrict and the bone marrow steps up blood cell formation in an attempt to keep the circulation going. However, the body can compensate for a loss of blood volume only up to a certain limit. Whole blood transfusions are routinely given to replace substantial blood loss and to treat severe anemia. The usual blood bank procedure involves collecting blood from a donor, mixing it with an anticoagulant to prevent clotting. The treated blood can be stored for about 35 days until needed. Although whole blood transfusions can save lives, people have different blood groups, and transfusing incompatible or mismatched blood, can be fatal. The plasma membranes of red blood cells bear genetically determined proteins, or antigens, which identify each person as unique. An antigen is a substance the body recognizes as foreign. It stimulates the immune system to release antibodies to mount a defense against it. The recognizers are antibodies present in the plasma that attach to red blood cells bearing surface antigens different from those of the patient's red blood cells. Now, binding of the antibodies cause the foreign red blood cells to clump, a phenomenon called agglutination, which leads to the clogging of small blood vessels throughout the body. This is something we do not want to happen. There are over 30 common red blood cell antigens in humans, so each person's blood cells can be classified into several different blood groups. However, it is the antigens of the ABO and the RH blood groups that cause the most vigorous transfusion reactions. The ABO blood groups are based on which two antigens, either type A or type B, a person inherits. Now, the lack of antigens is called type O. Absence of both antigens results in type O blood. Presence of both antigens leads to type AB blood, and the presence of either A or B antigen yields type A or B blood. Now here you can see which blood types can receive or donate blood. AB is considered to be the universal recipient because they can receive blood from anybody. O is the universal donor, and it can give blood to anybody, but it can't receive blood only from another O blood person. This table gives you an idea of the frequency the population has as to what type of blood. It also shows you which antigens and antibodies would be present in each of the different kinds of blood. Now, the RH blood groups are so named because of the 8 RH antigens that were originally identified in rhesus monkeys. Later, the same antigen was discovered in human beings. Most Americans are RH positive, meaning that the red blood cells carrying the RH antigen. Unlike the antibodies of the ABO system, anti-RH antibodies are not automatically formed and present in the blood of RH negative individuals. Problems can occur in mixing RH positive blood with a person who is RH negative. An important RH-related problem occurs in pregnant RH-negative women who are carrying RH-positive babies. The first such pregnancy usually results in the delivery of a healthy baby. But because the mother becomes sensitized by an RH-positive antigens that have passed through the placenta into her bloodstream, she will form anti-RH plus antibodies unless treated with something called RhoGAM shortly after giving birth. Now, this RhoGAM is an immune serum that prevents this sensitization and her subsequent immune response. If she is not treated and becomes pregnant again with an RH-positive baby, her antibodies will cross through the placenta and destroy the baby's red blood cells, producing a condition known as hemolytic disease of the newborn. Now, the baby is anemic, and brain damage can occur, and even death may result unless fetal transfusions are done before birth to provide more red blood cells for oxygen transport. Now, The importance of determining the blood group of both the donor and the recipient before blood is transfused is obvious. The general procedure for determining ABO blood type can be seen in this next slide. Essentially, it involves testing the blood by mixing it with two different types of immune serums, serum A and anti-A and anti-B. Agglutination occurs when red blood cells of a group A person are mixed with the anti-A serum, but not when they are mixed with the anti-B serum, and so on with the anti-B serum. So, what are the key points that we need to remember in this particular slideshow, our last slideshow? What is the difference between an antigen and an antibody? What are the classes of human blood groups? What is the probable result from infusion of mismatched blood? We know that. What is the Rh factor? And why is the Rh factor so important to pregnant women? This is all we're going to talk about when we talk about blood.